Hi, everyone. My name is Tim Traynor. I'm a part of the technical sales team and scientific support team at Alchemyᵀᴹ. It's a pleasure to have an opportunity to speak about Alchemyᵀᴹ and what's titled here, Driving the Next Generation of mRNA Manufacturing with Alchemy Cell-Free DNA and Codex HiCap T7 Polymerase. So in addition to talking about these products and services and how they service our mRNA manufacturing capabilities, I will also give an overview and introduction to Alchemyᵀᴹ and provide an update on the expanding products and services. So Alchemyᵀᴹ is a seasoned contract development and manufacturing organization, CDMO. We're proficient in supporting efforts from discovery all the way through commercialization. Founded in 1998, Alchemyᵀᴹ has amassed over 25 years of experience in providing our biotechnology and biopharmaceutical partners with the essential foundations necessary for advancing breakthroughs in the cell and gene therapy industry, as well as mRNA vaccines. So we are US-based operations founded in our headquarters, Fargo, North Dakota. And that's where our GMP manufacturing and research grade facilities, as well as R&D services, are there. We have our other innovation lab in Madison, Wisconsin, which is ISO 13485 certified and labeled our innovation lab, which provides ancillary raw materials, proteins for IVT enzymes, and gene editing nucleases for our GMP manufacturing site in Fargo. Historically, Alchemyᵀᴹ has established a strong foothold within the United States and North America, where our main U.S. operations obviously take place. Over the years, our reputation has expanded as we've addressed the European market, and we recognize the necessity to provide our European partners with the support they deserve. And in the recent years, we've taken significant steps to enhance our presence within that region. We've added not only just several commercial regional support representatives, but as well as some regulatory representatives on continent that have a good idea of the regulatory landscape over in Europe. Together with myself and the European team, our goal is to serve as a powerful conduit back to our facilities in the United States to ensure seamless support and collaboration. So highlighting our 25 years of experience, our products and service have evolved to cover a broad landscape of therapeutic and vaccine applications. And really, we have shifted our focus to these applications listed here on the left side. mRNA-based therapies, cell and gene therapies, gene editing therapies, and vaccines. And as you can see, we have a wide range of products and services that support each of those therapeutic modalities. And with this focus comes with our experience with over 1,000 advanced therapeutic companies supported worldwide. Of those, with hundreds of preclinical and clinical phases contributed to those companies, and as well as 15 fully commercialized cell and gene therapies supported. Of course, we're a proud collaborator with Moderna's COVID vaccine development and providing a linear template. From that, our manufacturing experience remains robust. Over 180,000 research-graded lots released. Over 11,000 of those are our clinical grade GMP source and GMP. And then as mRNA therapeutic landscape is kicked off, our manufacturing experience is increasing there as well. So the basis of our company was incepted in providing custom products and services. And currently, that's really the backbone, providing custom plasmid DNA, linear DNA, nanoplasmid, mRNA, and proteins. But as we've matured as a CDMO, Alchemyᵀᴹ has expanded a broad inventory of off-the-shelf products to simplify logistics and the supply chain as an accelerator to the clinic. And when we combine our custom manufacturing workflows with our at-the-ready, off-the-shelf products, we can create end-to-end workflows such as sequence to plasmid DNA, mRNA, and protein biologic, as well as mRNA drug product. In highlighting the multiple modality approach here, this graphic gives a nice picture of how our products and services support multiple different therapeutic applications. And as you can see, there's a nice overlap between modalities that you can see between plasmid DNA, mRNA, and protein products and services. And we continually refine our focus as the market continues to expand. So in summation on Aldebaran's overview, yes, we produce material across our three primary pillars, DNA, proteins, and mRNA. But at the core of our business, we offer essential solutions and products, including analytical assay development, process technology development, storage and stability services, comprehensive in-house QC testing, etc. And on the right side, right side of the screen here, around 3 years ago, we became a part of Danaher, and a growing list of companies there has effectively become the Danaher Life Sciences Group. And this group, growing network of companies, has one unified goal, which is accelerating the advancement of life-changing therapies. And Aldebaran is proud to be an integral part of the synergistic network within Danaher. And here we have a high-level overview of the genomic medicine journey of the Danaher that the Danaher Life Sciences Group focuses on. And together we're at the forefront of advancing genomic medicines encompassing sequence generation, delivery, manufacturing, and analytics. And our mission is to build a value-driven work stream that enhances efficiencies for our partners while upholding our unwavering commitment to quality services. And Aldebaran, along with IDT and Cytiva, is spearheading the development of these platforms and ensuring a comprehensive and integrated approach to genomic medicines. So moving on to the more core focus of this presentation, we're going to be this graphic shows our end-to-end mRNA drug substance and drug product manufacturing. And where I'm going to introduce our next product, I'm going to be focusing on the IVT part of the reaction of our mRNA-based services. So to address the primary concerns and safety costs, we have entered into a license for the HiCap RNA polymerase with Codex. And basically what it is a co-transcriptional capping mutant T7 polymerase that's engineered to produce synthetic RNA to meet the demands for improved safety and cost reduction. And the way that does this, right, is we see a reduce of the immunogenicity from double-stranded RNA byproduct and increasing capping efficiency, thereby increasing or decreasing the costs associated with mRNA manufacturing. So double-stranded RNA can elicit an immunogenic response and ultimately lead to cytotoxicity, as well as inhibit protein translation, which thereby can reduce the overall efficacy of the desired therapeutic outcome. And when we compared wild-type 7 HiCap to wild-type T7 polymerase, we see a reduction of the double-stranded byproduct of around 10x. And this is across both tri- and dinucleotide cap analogs, so ARCA and CleanCap. And we measure this by dot plot analysis. And the top two charts there show us the double-stranded RNA intensity via dot plot analysis, with the bottom quantifying the differences in mRNA input intensity with the cap analogs, as well providing, highlighting the cost and time savings with the optional removal of the final polishing chromatography step that is set up to remove the double-stranded RNA byproduct. And when we compare the capping efficiency between HiCap to wild-type T7 polymerase between CleanCap and ARCA, we see a greater capping efficiency. So specifically with CleanCap, we see on there 60 or a greater 95% capping efficiency, thereby requiring around 62% less capping reagent. And then for ARCA, same level of capping efficiency and requiring around 20%, thereby providing the more efficient approach to mRNA production with significant cost savings. And with the benefits of the capping efficiency and less double-stranded RNA byproduct, we also show that HiCap shows improved yields and maintain it across multiple different mRNA transcripts. So they're at 2 kilobases, 6 kilobases, and 9 kilobases. So overall, providing confidence across mRNA therapeutic modalities, and even with the larger mRNA-encoded nucleases that are coming to the market as well. So quick summary here, improved safety costs with HiCap generating significantly less double-stranded RNA, and the cost savings associated with the increased capping efficiency. And we also talked about the optionality or obsoleteness of that final chromatography step that's may or may not be needed, depending customers' therapeutics. And HiCap is currently available at a research grade and R&D quality level, and we're expected to roll out phase-appropriate grade material mid-2025 at our Madison Innovation Lab. Showing this graphic here again, we're going to shift our focus upstream on our mRNA products and services on the linear template side. And that segue is into introducing our most new product, Alchemy cell-free DNA. And what that is a cell-free process that enzymatically produces linear DNA template. And when we compared Alchemy to traditional E. coli-based platforms, Alchemy offers more than a 50% reduction in timelines by eliminating a lot of those standard DNA manufacturing steps that we see with cell-based platforms. Such things such as clone screening, different growth phases, and then multiple sequencing, QC confirmation steps in between each of those milestones. So how do we do it? We start from a plasmid DNA as the template and move into the enzymatic amplification process known as rolling circle amplification. And following this, we use the standard restriction, same standard restriction endonucleases to linearize the DNA template, and then followed by purification process and QC evaluation to prepare the cell-free DNA template for IVT reactions. And we see here that no optimization is required for mRNA production when we transition from traditional plasmid DNA workflows to the Alchemy cell-free process. And the way that we did this is we evaluated Alchemy cell-free platform against standard plasmid DNA platforms with industry collaborators. And some of the things they found to validate our cell-free DNA process, it demonstrate that the DNA template integrity is more or less equivalent or superior in some cases, with higher yields as well as stability in the mRNA product post IVT. This is obviously dependent on construct type, but here we see some DNA that shows these results clearly. The other thing they showed is the enablement of a consistent process for IVT production. And when they compared this to standard DNA templates, they saw lower residual DNA templates after treated with digested enzyme DNase removal, showcasing the outcomes that are comparable to or even better than the standard E. coli-based system. So consistent digest platforms, therefore, equals process consistency for our customers. And the features of the Alchemy cell-free DNA template is produces a linear double-stranded DNA template that is open-ended, eliminating the need for any additional capping or removal procedures prepared for IVT. And the sequence is identical to the plasmid DNA template and is specifically designed for mRNA production. So there is no, needed to removal of any homology or primers needed for IVT. It's standard across the production. And also the purification process, since it is a cell-free platform, a lot of the cell-derived impurities associated with those E. coli-based platforms are very minimal. So things such as host genomic DNA, residual RNA, host cell proteins, and endotoxin. So Alchemy is currently available at a research grade quality level, and we expect it to roll out the GMP offering by the end of 2025. And here is sort of that final integration step that I've shown before with our standard mRNA manufacturing process for drug substance and drug product. And here, as we integrate Alchemy into that mRNA process, we show that we have multiple points of entry for manufacturing. And that's what we really want to maintain as a CDMO, being versatile in production of what we can provide our customers. And with this, combining Alchemy and HiCap, we're pioneering the next wave of innovation of mRNA manufacturing services. And, we're able to do this with these products by reducing impurities, increasing efficiency, and deliver significant time and cost savings to our partners. And, before I close here, I know I spoke about Alchemy and HiCap or HiCap T7 polymerase, but Aldebaran has a comprehensive catalogs of other new products. And, like I said, touched on HiCap and Alchemy. But here you can see we have other newly innovative products that we brought to market. And if there are any questions, please invite everybody to reach out and get in touch with Aldebaran representative to help you answer more questions you may have.